Hi everyone, welcome to today's lecture. Um, this is not part of the exam two you guys are going to write to on Wednesday. I'm sorry, on Friday. This is not part of the exam two you guys are going to write on Friday. So do not learn this one. This is for exam three. So today we are going to talk about the immune response that we have, and we'll focus mainly on humans. So this is the learning objectives. Uh, We will summarize the three main responsibility of the immune system, and then we will list and describe the primary physical and chemical defense barriers on and in the skin, mucous membrane, and resident microbiota. We will state the two main body systems that contribute to immunity. We will differentiate between PAMs, PRRs, TLRs, and cytokines. We will name the type of leukocyte that function in the innate immune system and explain the most important functions of each. We will list the four major mechanisms of the innate immune system and describe the steps or stages or key features for each. We will summarize how interferons function in the innate immune system, and then we will compare and contrast the three different complement pathways related to activation outcomes and also the C3 converters. So, what is the purpose of the immune system? The immune system is basically responsible for the recognition of both self and non-self, with self referring to your own human body cells, that is the neurons that you have, the blood cells that you have, the smooth muscle cells, all cells in your body, and then the non-self referring to any foreign organism. The immune system is not only restricted to just defending your body, it's also responsible for cell communication. Today, we'll be focusing on the cellular and the molecular mechanism of the immune system as it relates to infection, which is the non-self. That can be any other thing beside pathogens. Excuse me. So, today, we will be discussing briefly the immune system in humans. But before that, I want to point out that there is immune system in different organisms. So, for example, in bacteria, we have the CRISPR-Cas9 defense system. I'm sorry, the CRISPR-Cas defense system, basically it protects the bacteria against bacteriophage by cleaving it and cutting it into pieces, cutting some DNA sequences in the bacteriophage into pieces. And then we have the plants immune defense, which is basically two branches. This is actually similar to the immune system in animals, but not mobile cellular constituents like the one that we have. And then in animals, we have innate immune system, which is present in all animals, and we have acquired and adaptive system, which is only present in vertebrates. So as I mentioned, today we'll be focusing only um we'll be focusing only on the human immune system. So in the human immune system, there are two main branches. We have the innate and the acquired. The innate having subcategories, which is the barrier defenses, and then the innate immune system, which is more cellular. And then we have the under the acquired, we have the humoral immunity, which is mostly by B cells. And then we have the cell-mediated immunity, which is also done by the T cells. So first, let's talk first about the barrier defenses that we have. So under barrier defenses, this is basically your first line of defense all over your body. And the goal is to prevent any invading microbes from getting into your body, and is one of the largest physical and the chemical barriers that we've known. There are three major barriers under this. We have the skin, which we have all over your body. We have the mucous membranes, which is present both in the eye, the digestive tract or your oral tract. And then we have respiratory, we have genitourinary also tract. And then lastly, we have the resident microbiota, which is present in your gut. So you do realize that throughout the whole body, there's almost every part of the body, there's a defense mechanism there. So first, let's talk about the skin, which is more visible on your body. You can see it. It's a thick, tough, waterproof layer covering your body, which is mostly keratin, is mostly made up of keratin. It's very high in it's very low in acid, and in acidic, the pH is very low, so basically it's acidic, and it contains fatty acid content. And in it, we have your hair and your sweat, which is responsible for getting rid of preventing most of microbes invading your body. And we also have lactic acid and electrolytes in your sweat, which is responsible for making the environment uncomfortable for any invading bacteria. And lastly, we have ear wax, which is responsible for preventing the movement of bacteria into your ear. Because the wax is thick, it just prevents the movement of bacteria into your ear. The second one we'll be talking about under physical barriers is your mucous membrane. These are present in both the digestive, genitourinary tract, respiratory tract, and also the eye. And their main mode of defense is secretion of mucus coat, which impede the entry and attachment of bacteria. And then they also have mucosal secretion, which contains antimicrobials, basically fighting against the invasion of microbial organisms. So to take a focus on the eye alone, you do realize that sometimes you feel like something just got in your eye and you begin to tear, or you blink and then there's tear coming. The whole concept is that whatever invaded your eye or whatever fell on your cornea, it just triggers an immune reaction which results in you blinking and tearing up. But within your tear, you have these enzymes we call lysozymes, and basically, they just hydrolyzes peptidoglycans in the cell wall of the bacteria and just go ahead and just like here, they go ahead and just cleave it and get rid of the peptidoglycans in the cell wall, basically disrupting the bacterial cell and letting the bacterial cell die. So, another mode of you secreting mucus is through saliva, basically liquid fluid flowing out of your mouth. And then within this fluid contains lysozymes. And then their goal is again to disrupt the peptidoglycans of the bacteria. And also within your stomach, we have stomach acid, which basically prevent the survival of microbes, which will even escape. I'm so sorry. Uh, which will even escape your mouth and then get into your tract. And even within your digestive tract, we have bile and also digestive enzyme, which main focus is to kill any bacteria that will also get in there. So, as much as you have these mucus also in your mouth, we also have some present in your respiratory tract. So the first one is you have nasal hair present in your nostrils, and basically the whole goal of your nasal hair is to trap larger particles, preventing any larger particle going to your nasal tract. And even if some particles bypass this nasal hair phase of defense, which is basically present here, there's also mucus, which is more present into your nasal tract and basically traps and flushes out any microbe that you have. And then let's say it also get the bacteria get away with this. They are also ciliated epithelium of your airways. Basically, here where you have some thick epithelium cell present with ciliated or more like a meshy nature of it. And its role is to just also helps in the movement of mucus and trapping of any other bacteria that will even invade both the first and the second mechanism I just explained. And then within your genitalial tract, uh, you experience a continuous tickling of urine through your uterus or your bladder or your female part. And then basically the goal is to try and flush out any bacteria that might be present. And also in males, we have antimicrobial component within your semen, which is responsible for getting rid of any microbes that will be present. And also in females, because of the acidic nature of your genitalia, it just prevent the growth of microbe present down there. So it's just basically the place is super acidic, preventing the normal growth of bacteria. So, and the physical barriers, we also have your resident microbiota. So basically, like we said, in every human being or in all of us, we all have gut microbiome. And then these form like as in a niche exclusion of physical barrier, and their goal is to exclude pathogens by outcompeting them. So you already have microbes within you, they are just outgrowing the pathogens that will get into your tract, your digestive tract, and basically they will create an unfavorable environment for pathogens. So an example is this. This will be probably like your gut microbiome, and this is the pathogen invading you, and it's just fighting it to get rid of it. One example of ways for this gut microbiome to outcompete the pathogen is like with um, E. coli synthesizing colicins in the intestinal tract, which will basically make the environment unconditional for any outside microbe that is present. But the main point you have to pay attention is to that when your gut microbiome lose this fight or they lose their function, it will lead to super infection, as we discussed from our earlier class, that super infection will result in the pathogens outgrowing your gut microbiome, and then you get infected. So as we normally do, I'll leave this for you guys to practice, and when we meet also in class, we will talk about this more in details, and we will communicate and share ideas within our groups. But these are the answers. So now let's move to one. Now that we are done with physical barriers, let's move to the innate immune system. So under the innate immune system, we first have to talk about the circulatory and the lymphatic systems. So what is the circulatory system? Basically, the goal is to just imagine it as your blood system. So basically, your goal is to the goal is to circulate immune cells and factors all around. So now you have your blood is just circulating immune systems all around your body. And then once you have blood, you have red blood cells in there. So they are basically also circulating oxygen, nutrients, and also hormones to all the cells that are present, and they aid in the removal of waste. So this figure shows the diagram of a circulatory system all over your body. And then next to it is the lymphatic system. So the lymphatic system is part of the immune system. Their main goal is to contribute to the maintenance of bodily fluid. So just as the blood system, the circulatory system is circulating around immune cells and providing with oxygen and other things, the lymphatic system is maintaining your bodily fluid, making sure you have enough fluid in your body, and it aids and it also helps in the defense mechanism by absorbing digestive tract fat and the removal of cellular waste, which you already have in your body. And then eventually, okay, so now let's move to how the communication occurs between body compartment. So the immune system is largely a cellular and a chemical type of mechanism, and then they contain cells and signals that move throughout your circulatory system and lymphatic system, and also within tissues. And as you can see here, you have your immune system being present. The circulatory system being present, and they are communicating with outside of the tissues and cells outside of of the vessels. For example, you can clearly see there's a macrophage present here, and then we have the extracellular fluid. So you can clearly see there's a clear communication between the immune system and the lymphatic system. But today we'll be focusing on the cellular and molecular mechanism of the immune system as it relates to infection. So let's talk first about the immune system responsibility. So let's talk about the immunity responsibility. So under innate immunity, basically what it does is recognizes self, basically your self cells, the cells within you, and the non-self, which is the extra pathogens or the outside pathogens coming within your body. It just detects them. Um, it helps in the detection of a broad non-self molecular molecules of microbes. So these microbes or these microbial organisms are being classified as PAMs. And I'll talk much more about what PAMs are. But basically, your innate immune system recognizes this, and then what they do is they serve as a cellular communication by secreting cytokines in cytokines, which will then attract or communicate with your immune system. And then lastly, it will result in a destruction of the foreign material or the non-self that it has first identified through either phagocytosis, antimicrobial proteins, fever, or inflammation. So just a short recap. This will be your innate system, like your security cameras. They see the invaders, and then what they basically do is just call or phone and warn that there is an invader present. And then there is a security team being sent into it, which will be discussed much more. Can be your white blood cells coming in with phagocytosis, coming to get rid of anything present. So as I mentioned earlier, the innate immune system helps in the recognition of PAMs. So what is PAMs? PAMs basically means pathogen-associated patterns. So these are molecules which are recognized by phagocytes and some other cells. They are typically found on the surface of microbes, and they are molecules which are not present in mammals. So the foreign organism or the foreign pathogen which is coming within you have other molecules present on their surface. These molecules or these surface proteins are not present already within us. So it's easy for the immune system to recognize this, like this is not part of us. So then it means it's a foreign organism. Some of the common microbial features, which are not species specific, include peptidoglycan, which we already know is present only in bacteria. We also have double-stranded RNA present in virus. We also have lipopolysaccharides, which are LPS, which are only present in gram-negative bacteria. So, another thing that the non-self how another thing that helps in the recognition of non-self also is the pattern recognition receptors, which are PRRs. They serve as surface receptors, basically serving as the eyes and the ears of the system, and they help recognize and bind directly to palms. So your cells will have basically constitutively expressing always on. So what you have is your cell always have these PRR, which are basically pattern recognition receptors. So now the immune cells have pattern recognition receptors. Some are in the on the surface of the cell, some are within the cytoplasm, some are present on endosome and also on phagosomes. And their goal is that they are the security cameras, so they are always on trying to scout for external factors or external pathogens which will be invading your body. So under these, we also have toll-like receptors, which are just the subtypes of the PRRs that we just previously discussed. We have over 10 toll-like toll-like receptors present in humans, and they are on cytoplasmic membrane of our cells, and they monitor extracellular movement, basically making sure that no known pathogen is being unrecognized. They make sure they recognize everyone and just signal the immune system. And they are basically always available to monitor ingested materials through either endosomes or phagosomes. So these are some examples of PAM and PRR interactions which we know. So if we take an example, a viral a virus here, and you can clearly see this is a viral capsid. And then we have the toll-like receptors, the TLRs, recognizing the surface glycoproteins which are present on the virus. So they can recognize TLR2, TLR4. And then the list goes on and on. We also have some surfaces, some TLRs recognizing TLR3, which is within the virus, and TLR7. you can just go through the whole list. But basically, the concept is that we have PRRs, which include in addition of TRRs, and they recognize PAMs, which are patterns which are not present in your body. This is only present in the pathogen, and then the PRRs in your body are recognizing them. So the immune cells are not only there to recognize the palms which are coming from the pathogens, but they are also present for communicating within each other. And then basically they do the communication through two mechanisms. The first one is through the effector function, or basically the function of cells relating to destroying non-self. And the second one is the secretion of cytokines for communication. So let's say this macrophage, it already have the toll-like receptor to recognize something, and then it secretes cytokines or anything, and then send the message to the nearest macrophage for it to recognize what is going on. So sorry, I think I explained the opposite way. So now this macrophage recognizes the foreign material and secretes cytokines to this macrophage, and then this macrophage then move closer to this cell. So this mode of communication allows for coordinated responses between those two cells, and all of not only these two cells as an example, but basically throughout all of the cells present in our body. So now let's talk about the mode of communication which we've seen. The first one is cytokines. Cytokines. You can just see these as your voices, or like the way we shout and just talk to someone next to the table or someone in the room. Cytokines, you can just imagine them as voices. These are basically secreted proteins from a cell, and these proteins are being secreted and moved throughout the whole cytosol. And basically, there's also a target cell which is having the cytokine receptors. And these receptors, the cytokines bind to the receptor, signaling the other cell or whatever communication the first one is communicating to it. So that's basically how it works. So it's just been like this cell voicing out whatever you want to say through cytokines to the other cell. But what you have to know is that this works locally and regionally, and it's also going to work systematically. So it wouldn't work throughout the whole body, but it's going to work locally depending on where the cell is being found. So what are some of the type of cytokines involved in immunity? So we have pro-inflammatory cytokines, which are basically used to promote inflammation and fever. An example is interleukin 1, we have TNF. And then we have chemokines, which are basically chemoattractants, which stimulate the migration of other cells. So, for example, let's say these cells secretes chemokines to this cell. Basically, when this cell receives the signal, it starts moving in the direction of this cell. Because probably, let's say this cell have found a pathogen present and just secrete like, hey, there's a foreign guy here, can you come get rid of it? Then this cell start moving in the direction of that cell. We also have vasodilated and vasoconstrictors, and basically their own function is that they change the dilation or the diameter of the blood vessels, either permitting permeability or just decreasing permeability. And this is mostly involved when it comes to fever, and we'll talk more about that. So the third one we will have to talk about is the distraction of the non-self. So now we've talked about recognition, we've talked about communication. The third step is to talk about the distraction of the non-self. So these depend on the cell type, and it's not pathogen specific, so it broadly target all range of pathogens. And we will discuss four main mechanisms after we talk about this cell type. The first mechanism is phagocytosis. The second mechanism is fever. We have inflammation and antimicrobial proteins. These are really like at the end of the lecture, so we talk about it later on. So before we jump into the other part of the immune recognition and response, we have to understand that under these and under this under this broad range of non-self-recognition, we have an important type of cell type, important types of cell types that are involved in this mechanism. These cells are present in the blood. So what these cells do, what group of cells do this? These are called leukocyte, or basically white blood cells. And under white blood cells, we have agranulocytes. And basically, they consist of monocytes, which will later turn into macrophages and dendritic cells. We have T cells, we have B cells, we have natural killer cells. Don't try to memorize every single thing. I'll go through later on in the next few slides. And then we also have non-leukocyte, which are basically red blood cells and platelets. And then we have granulocyte, we have neutrophils, basophils. All of these we'll go through in the next few slides, one after the other. So now, first thing that we have to talk about is the groups of white blood cells and what they do. So under this, we'll first tackle the granulocytes, which are basically lobed nucleus with they are basically seen as a loop nucleus with cytoplasmic and antimicrobial granules, as you can see basically within them. And the subgroup of these white blood cells are neutrophils, eosinophils, basophils, and mast cells. Then we have another class of white blood cells, which are agranulocytes. These are unlooped. They are rounded, they have rounded nucleus with no antimicrobial granules. And these are lymphocyte B cells or T lymphocytes, and we have monocytes, which consist of dendritic cells and macrophages. So now let's take each one after the other. First, we will talk about neutrophils. So what are the function of neutrophils? Neutrophils basically engulf or phagocytose and destroy bacteria. One example is being seen in this image, where we have our bacteria present and we have neutrophils going here and just eating them up or engulfing them and destroying them. And then you can just imagine them as the SWAT team, where they come in and just clear the whole place. So imagine you have this amount of bacteria cells present, and you just introduce neutrophils. Within a matter of short time, they just clear everything all out. So they can increase to higher numbers during infections, and they are just short-lived. So they don't stay there for a long time. They just get the job done and they just die off. So you can imagine them as a SWAT team, just rapid response, swamps in and just eliminate. And their main goal is to just kill by phagocytosis, basically engulfing or eating stuff. They are mostly present and they circulate around 6 to 12 hours, and they are about then they just get the job done. Within 8 days, that's their short, they have really have a short life cycle. So within 8 days, they just die off. But their mechanism is super swift. Within 6 to 12 hours, they just get the job done. The next group is eosinophils, and their function is specialized attack and destroy eukaryotic pathogens. So they also contribute to response in allergies and inflammation, and they are mainly through release of content in granules and also some phagocytosis. And as you can see, they are also doing a similar job. They come in here and go off everything and just get rid of it. They arrive super early during infection or allergy site, and they consist of about 2 to 4% of circulating white blood cells. The next one is basophils, and their function is related to they being present during allergic reaction or inflammation. And you always find them in an environment which is being inflamed. They are they contain granules. They are granules and they contain histamine, mainly one of the major chemical modulators causing allergy symptoms. And their granule content are being released out in the environment. They share similar morphology and function characteristics with mast cells, which we'll talk about later on. So the next one is mast cells, and they are found in tissues, not typically circulating, but most often at the mucosal surface and the epithelial surface. Their function is also similar. They come in and just engulf any other pathogens that we can see. They are mostly triggered in localized inflammation, and they are granules, and they also contain histamine. So now that we've talked more of this, we will discuss these in acquired immunity lecture. So we won't spend much more of the today's lecture on T cells and B cells, but we talk about this in immunity. But let's first talk about the other subgroup of agranulocytes, which is monocytes, which will later grow into macrophages and dendritic cells. So what are monocytes? Monocytes are these cells that have granules, as we discussed earlier, with digestive enzymes in them. They are initially born in the bone marrow, and they will later migrate and move into the bloodstream and end up circulating. They rapidly leave the bloodstream and just matured into macrophages and dendritic cells, as is being shown down here. They are the largest of all of the white blood cells, and they live as phagocytes for a few days in the bloodstream and then go to the tissues as macrophages. So when they leave the bloodstream, they will leave the bloodstream during circulation to mainly reside in tissues, as we've been saying. They live for about weeks to months, where their major role is to just monitor or patrol throughout the whole tissue. And they come in many types, and for that reason they have different names based on where they are being found. For example, we have alveolar macrophages, basically because they are present in the alveolar and their own alveoli. They are present in the alveoli in the lungs. And then here we have Kupffer cells, which are which are always present in the liver. And then we have osteoclasts, which are present in the bone. And as you can see, here is an example of macrophages being present in the tissue, and then they are just doing their job, engulfing everything that they can find. And then phagocytosis. So what is the function of macrophages? Their function is basically to phagocytose all pathogens that they see. Basically, they are just getting rid of all the messes which are being caused by inflammation and infection as they patrol the tissue. And then the second one is to communicate. So what they do is, after they've phagocytosed the tissue, they secrete chemokines to recruit other immune cells for them to come to the area. And they also process the foreign molecules that they have engulfed and present it to lymphocytes. As you can see from here, this is macrophage, takes out the foreign cells and then secretes chemokines and then present this to lymphocytes. Next on our list is dendritic cells. As I said earlier, you have mac you have monocytes grow and become macrophages and also dendritic cells. So now we've discussed macrophages. Now it's time for us to discuss dendritic cells. They have unique long protrusion called dendrites, hence their name dendritic cells. They travel to form peripheral tissues. They travel from the peripheral tissues to spleen and lymph nodes, and their main function is similar to the macrophages. As you can see, they are responsible for phagocytosis of pathogens, and they also are involved in communication. So macrophages are more present in the circulatory system, whilst the dendritic cells are more present in the lymphatic system. So, I'm so sorry. I'm sorry, guys. So back to communication again. So they process foreign molecules similar to macrophages and present it to lymphocytes, specifically T lymphocytes, and they also secrete cytokines to recruit lymphocytes and other immune cells. They are extremely better at activating T cells than macrophages. So let's practice again. We have neutrophils which come in as the SWAT team. And then we have macrophages, which are always policing around like normal police officers. But the neutrophils just comes in within a short time, get rid of what they have to get rid and just go. And then we also have eosinophils, which comes in as the special forces when it's extremely needed. And then here you can think of these guys as basophils as the disaster response team. When there's something extremely present, then the basophils comes in. And then we have the dendritic cells, which are more surveilling as the CIA. And then we have mast cells, which are basically securing security tower guards. So now let's talk more about the innate response. So we first have discussed we have first discussed about the recognition of self and non-self recognition of palm, and now we discuss innate response as their one important function is for them to help in communication through cytokines. Now the third one for us to talk about is the destruction of foreign materials, which is the non-self, through phagocytosis, antimicrobial proteins, fever, or inflammation. So we take this one after the other. We first talk about phagocytosis, inflammation, fever, and then the last one will be antimicrobial proteins. So phagocytosis. What is phagocytosis? Basically, cellular eating or just a cell eating any foreign material that it finds. And this is basically a type of endocytosis or just engulfing of outside things into outside material into the cell. So the phagocytosis, basically their role is to phagocytose for a living. So they just have to eat something or any foreign organism in order for them to survive. And their general activity is just to survey. You survey, you find microbes or particles, and then you find this mostly at injured sites, or you can also find a dead cell and just ingest it and eliminate it. And their role is to recognize immune informations or the palms, and then take control of whatever happens. Take control of the system that they find themselves to, the situation that they find themselves to. That is to get rid of any of the foreign organism present. So now this is a summary of phagocytosis. I'll discuss a short summary, but we go very deep into each step. So um, imagine this is a macrophage, and basically the role or any of the white blood cells. Let's take this as a white blood cell in general. So any of the white blood cells, this is it, and then this is bacteria present in your blood, and then your white blood cell have these PRRs that we learned earlier on. These are the receptors. They recognize the bacteria, and then once they'll be able to recognize the bacteria, which is in our case the palms or the external pathogen, have to bind to the receptor on the white blood cells. And then once the white blood cells signals or recognize this foreign body, it basically forms this engulfment. And once it engulfs it, it brings it within the white blood cell itself. And then what basically happens is that the white blood cells secrete lysosomes, which is going to come from the Golgi apparatus, and then the lysosome comes in, and also basically the lysosomes are enzymes that are going to just degrade or cut the bacteria into pieces. So the lysosome comes in and enters the bubbles or phagocytosomes that it have already been formed. And then the lysosome just help cut the bacteria into smaller pieces. And after cutting it into pieces, it just engulfs it's just like it engulfs everything and just brings everything, releases everything out of the cell. So basically, that's what I just described. That is the whole process of phagocytosis. I'll be moving on and describe and discussing every single step into details. So the first two steps are chemotaxis and recognition or adhesion. So basically, there have to be a signal of chemotaxis which are being produced by the microorganisms that invade your body. And basically, these are secreted by the bacteria, and sometimes you can also secrete this from cytoplasmic membrane or phospholipid from injured cells. So some of the injured cells within our body also secrete chemotaxins. And basically, what they are secreting are chemokines, which are secreted to the immune cells, telling me, hey, something is wrong. And then once the bacteria, which is secreting, in our case, let me use the bacteria to make it simpler. So in our case, once the bacteria secretes the chemotaxis or the chemokines from the bacteria, and now the immune cells have to have these PPRs or the pattern recognition receptors present, and they have to recognize the message being sent by the foreign bacteria or the palms, and then it have to recognize and attach the palms attached to the palms through the PPR. So now your immune system, sorry, your white blood cell have to bind to the bacteria through this receptor. So now it has now established an adhesion with the bacteria. So first, chemokines have to be secreted, or chemotaxis have to signals of chemotaxis have to happen, and then next is the attachment of the immune system to the bacteria or the foreign pathogen. Following that, the bacteria, sorry, your immune system, your white blood cells have to then engulf. So basically, it just engulfs the pathogen that it have. And then once it engulfs it, it forms these phagosomes, which are just basically vesicles wrapping around the bacteria within the white blood cell itself. And then they have to need to a secretion of the secretion of lysosomes from the Golgi. So this stage I just described is the engulfing or ingesting stage and the generation of phagosomes, which is this. So the next stage is the phagolysosome formation and the killing. So now, as I described from here, phagosomes is being formed, lysosome is being released. So now the lysosome then comes in and then it fuses with the phagosome. So lysosome comes in and then fuses with the phagosomes, and then it releases the antimicrobial chemicals and also the enzymes, which are phagocytes, into the releasing of the phagosomes into the oh no, releasing of the lysosomes into the phagosomes, and then the lysosome goes in there and then a size or cut the bacteria into pieces. So lysosomes or the chemicals and the enzymes released by the lysosomes is contributing to both enzymes, or it can also be in the form of reactive oxygen product. It can also be in the form of lactic acid, which is contributing to low pH. It can also be in the form of peroxidase, which contribute to the production of toxic oxygens. But basically, well, it leads to is respiratory burst due to reactive oxygen species being going in there. And the whole goal is that you go in there and just disrupt the whole bacteria cell, break it down into pieces. And then in the next stage, it just releases it out. Now, let's talk more about phagolysosome formation and then killing. So there are different mechanisms that results there are different mechanisms that the phagolysosome takes. Sorry, I think let me connect back my screen again. Okay, so there are different mechanisms that happen for the disruption of bacteria to happen. The first one is NADPH oxidase, basically a protein complex that generates reactive oxygen species. This we discuss mostly in our class. So we have NADPH, which oxidizes, releasing NADP and oxygen. And then we have the oxygen moving into the cells to form hydrogen peroxide, and then or forming or super oxide, and then this is down further breakdown into HOA, and then this goes in there and then describes the whole cell wall of the bacteria. The second mechanism is the vacuolar hydrogen ATPases. Basically, they affect the pH in the phagolysosome, contribute to phagosome fusion. So as you can see from here, we have the injection of hydrogen ions outside of the phagosome into the phagosome itself. Now you have the releasing of the hydrogen ion, changing the pH, making it more acidic. The hydrogen ion will also mix with this oxygen, contribute to superoxide formation, both of which leads to the destruction of the bacterial cell wall. And then within the phagosome itself, we also have antimicrobial peptides, which goes in and also disrupts the bacteria. We also have proteases, which breaks down the bacteria itself. We also have degradative proteins, also breaking down the bacteria. And one outside mechanism that also happen is the transport of metal transport is the activation of metal transporters. This happen through lactoferrin and metal transporters. Basically, what they do is that they decrease ion availability, which is being used by the bacteria. So lactoferrin helps pump out ion through the metal transporters and get rid of all ions present in the lysosomes. The last mechanism is the the activation of ionos. Basically, they produce nitric oxide that damages the DNA and it activates protein. So nitric oxide is produced by ionos, and then nitric oxide moves inside the bacteria and then moves into the bacterium and just disrupt all the bacterium DNA, preventing it from replicating or making it more of itself. So this is how complex all these mechanisms are happening as a result of the release of lysosomes and then contribute to phagocytosis within the phagosomes. Last one is the release of debris. So after everything have been destroyed, the bacterial cells have been destroyed, DNA, everything have been destroyed, debris are just released outside the immune cells. Typically, it is done through exocytosis, and then the whole bacteria is just released out, leading to us being fully healed or prevented from infection. So this is also there for you to practice. As you always know, we will discuss more of this in class, and this is the answer to it. So we've talked about phagocytosis. Now let's talk about inflammation. So inflammation is just basically you seeing a sudden swelling or redness of your skin, and there are four classic signs of inflammation. The first one is rubor, which is redness or increased circulation or vasodilation. The next one is color, which is warmth, increase in blood flow. And the next one, third one is tumor, which is swelling or increase in fluid escaping from your tissues. Don't worry, we'll go deeper into each one of them. The fourth one is dolor, which is pain or stimulation of a nerve ending. And then the fifth one is just loss of function. So you can clearly see that the rubor comes with redness, tumor comes with swelling, dolor with pain, and color with heat. And all of these just happens based on you having inflammation. These are basically subclasses of inflammation. So now let's talk of the major functions of inflammation. The major function of inflammation include to recruit immune components to the site of injury, to repair tissue damage and clear away harmful substances, and to destroy microbes and block their further invasion. So inflammation can be broken down into early, middle, late stages, which are typically outlined as 4 steps, and we'll talk more about this later on. But the main goal is that inflammation is helping recruit injury and just prevent or destroy any other further microbes present. So let's talk about the first one, which is injury. So in injury, In injury, we have resident macrophages or other sentimental cells that will go ahead and detect pathogen. So now we get injured, and then we get injured, and then we have okay, let me take this again. So right now, what I'm doing is in our next few slides, we'll be talking about the process of inflammation. So what happened before you get inflammation and the whole mechanism of inflammation. That's what we'll be talking about. So the first one is injury. So now you get infected or you get injured, then the resident macrophages or the cells there will try to detect the PAM or the pathogens which are present. They are doing this through these PRRs, which are the receptors present on your cells, and the PRRs have to bind to the PAM. So now, after that connection have been done, we now move to the chemical signal release, which is release of pro-inflammatory cytokines, either through interleukin 1 or TNF. And the next one that will follow is the secretion, the activation of vasoconstrictors or vasodilators, and then secretion of chemokines. So as in these figures, you can see, let's say you get a needle pierce your skin, and then bacteria is just released, the contaminated needle have bacteria on them. What happens is that there's an immediate recognition of palms, and then there's a signal of your immune cells through secretion of chemokines. So the next step that we have to talk about is these two. So now we've established it, we've recognized the palms, we secreted the chemical signals. Let's talk about we've secreted the chemical signals through release of cytokines. So now let's talk about vasoconstrictors and vasodilation. So now you've secreted everything, everything is sent out, communication has been sent. Then what happens next is vasodilation. So vasodilation is good, and it happens for main reason is for recruitment of the cells. So you have to recruit cells, your immune cells have to be recruited to the site of injury. And this whole mechanism happens because your body your body activates this vasodilation by increasing blood flow at the site of injury. And this is basically what you see outside as heat or redness that you can feel in your body. And what happens is that the in nature or naturally, the blood vessels are more tightly constricted, and then blood flow or fluid flow from inside the blood and outside of the blood is being super regulated. But during vasodilation, this flow is loosened to in order to help your immune cells move from within the blood to outside to the site of injury. So the leakage of the fluid which you are being seen on your skin contains antimicrobial products and fluid dilute toxic substances, which are just present to get rid of all of the bacteria being present. So as you can see, the immune cells are now leaking out or moving out of the blood vessels to come and attack this. The initiation of vasodilation is basically done by chemical attractants or mediators, and mostly also done by cytokines. So if you are injured, clotting will definitely occur, and then thirdly, to be followed by the recruitment of neutrophils and other white blood cells to arrive through chemokines and then get rid of the bacteria. So one thing that I pointed earlier on is the movement of the blood cells through the blood vessels and coming outside, which is being termed as diapedesis. So basically, movement of leukocyte or white blood cells from the bloodstream into the tissue, and these are actively done by the leukocyte or the blood white blood cells are actively motile, and they have the ability to kind of change shape. So here they are round, and you can clearly see they are changing shape just to squeeze their way throughout, and they are doing this in the direction of this chemo attractant which has been secreted from the side of injury. And then you have endothelial cell receptors, which basically capture this cell and they participate in the transport. So these are the endothelial cells. These cells here are the endothelial cells, and they just vasodilate or just help in the movement of the white blood cells outside of the blood wall, of the blood cell, of the blood vessel, not the blood cell, of the blood vessel. So what will happen as a result of vascular leakage or vasodilation leading to this vascular leakage? And what we normally see is edema or swelling, which we see outside of the cell, outside of our skin. So as you can see, the exudate, which contains the blood-borne pathogens, which contains the blood-borne component that escape into the extracellular component. So now the that escape into the extracellular space. So now the blood-borne component, including the white blood cells and any other antimicrobial things which are being secreted from the blood cells outside of the tissue, um, basically moves all to the side of the injury. And basically what we see is just this inflame or edema that swelling or edema that we see. So edema basically means local swelling or firmness due to accumulation of the exude into the tissue. So the exude is the stuff which is being secreted from the blood vessel to outside of the blood vessel to the side of injury. And then the pus or the pus or pus, depending on who is saying it, is the whitish mass of cell liquefied whitish mass of cell, liquefied cellular debris, and also the bacteria. So everything which is kind of mixed out together is the pus. So the fourth step that happens is now we have inflammation or we have edema and everything comes out. Then you have the white blood cells present. They are getting rid of everything. You have to lead to a site which is called resolution, where everything has been done. So after the macro after inflammation have been done, what happens is macrophages are being attracted, and then macrophages comes in and that they clear up the pus, the cellular debris, the dead neutrophils, the damaged tissues. They get rid of everybody. And then the B cells and the T cells are also recruited to help clear the infection. So at the end of the day, you end up having a scar, and under the scar, you have your lymphocyte and your macrophages present to clear out all the bacteria and get rid of everybody, which results in the resolution of the edema, which was already being formed. So just to wrap up what we just learned, first one is you have an injury and you get an immediate reaction, which leads to the secretion of chemokines, and then these chemokines result in vasodilation of your blood vessels, which help in the release of your white blood cells outside to come and battle the external pathogen. This will result in edema because most of the fluid and the blood cells are being leaked out and they come all outside, forming edema and pus. And then lastly, after infection have been after the bacteria have been defeated, macrophages before the bacteria be defeated, macrophages and the lymphocyte have to be recruited, and the macrophages just come in and just engulf the bacteria and just get rid of them and just cut them into pieces and get rid of them. So now we fully understand that under the four major mechanisms of innate immunity, phagocytosis have to happen, and then we have inflammation, which we just discussed, and the last thing, the third one is fever. So I know when I say fever, everybody will be like, "Oh, what's happened? Why do we need fever?" Fever is actually a good thing, and we'll discuss more why fever is actually a good thing. So fever is nearly universal symptoms of infection. So almost all infections will result first in fever. It's associated with certain allergies, cancer, or other organic illness. So these are the ranges of fever. We have low fever, which is about 100 Fahrenheit to 101 Fahrenheit. And then we have moderate, which is 102 to 103. And then we have the high, which is 104 to 106 Fahrenheit. Fever basically just means an abnormal body temperature. So what causes fever? Fever is caused by pyrogens, which is basically any external or internal signal that is released to communicate with the thalamus to regulate your body temperature. So pyrogens are being secreted or pyrogens are being communicate through two forms. We have the endogenous communication, which is produced by your body itself. It's just released by monocytes or neutrophils and also macrophages during the process of phagocytosis to tell your body to just heat up and make the place uncomfortable for any foreign pathogen present. And this is mostly done through cytokines such as interleukin and TNF. And then we have exogens, which are produced by the bacteria, and they are products of infectious agents such as endotoxins, blood products, vaccines, or any other thing which is foreign to your body. So what happens is, for example, here you have exogenous pyrogens, which comes, they are already in the tissues, and then they will secrete they will be secreted into the blood stream, and then they goes to the thalamus and tell it, hey, make the body hot or make the body cold. And then we also have your endogenous pyrogens, which are being secreted. They also tell the thalamus to do exactly the same thing. So what are the benefits of fever? It inhibits multiplication of most pathogenic microbes, basically making the body unfavorable for them because it's too hot. And then it impedes bacterial nutrition by reducing available iron. As we discussed earlier on, the body will try to pump out the available iron through ionos and get rid of all the ions that can be used by the bacteria. And three, it also increases rate of host enzymes. Host enzymes, basically through metabolism and immune reactions. So, should we suppress fever? Slight to moderate fever, there is a potential benefit and minimal side effect. But if there is a high and prolonged fever, then that's actually bad. And so this is just an illustration. This is your optimum body temperature. And if you have a psychrophile, optimum body temperature, you have psychrophiles who will survive at this rate. And we have the thermophiles who will also survive at this rate. And then let's say the organism you have is present use maybe used to this optimum temperature. Once you have fever, and you have high temperatures, that organism is bound to dying because the environment becomes non-normal for it. The environment is not normal for such an organism, so the organism will end up dying. So the last part to talk about is antimicrobial proteins, which have three major types. So the first one that we have to talk about is antimicrobial peptides. These are short proteins capable of inserting themselves into the bacterial membrane. Over 5,000 of them have been discovered or created. They are basically about 12 to 50 amino acid in length. They are often cationic and also they have a hydrophobic portion. Their main goal is to just basically disrupt the bacteria cell membrane. So you have them here, they come in, disrupt the bacterial cell membrane and leading to the bacterial cell death. We also have some that target viruses and also eukaryotic microbes. They are mostly present in phagolysosomes. So we discussed earlier that the lysosome will secrete a lot of microbial peptides or proteins just to help degrade the bacteria. So as you can see from here, we have a positive we have the peptide which have the positively charged portion which helps attaches it into the bacterial cell membrane, and we have the hydrophobic portion which just basically engulfs into the cell membrane, degrading the cell membrane, causing a cellular burst of the microbe or the bacteria. So we have three different types. We have demicidins, which are basically a family of peptide. They can be cationic, they can be amphipathic, and they can also interfere with protein synthesis, directly damaging the DNA of the bacteria. We have proteagrains, which are basically family of peptide. Also, they can be cationic, they can be amphipathic. And then we also have catalycidin, which is also an example, and we also have the demicidin, which is also an example of it. All of these does basically the same thing, attached to the cell membrane of the bacteria and just degrade it. The second group we have to talk about is the interferons. So these are chemokines that causes neighboring cells to express antiviral proteins. So they are produced by certain leukocyte and also by infected tissue cells. They are non-virus specific, or they are not virus specific, so they can detect viral RNA. These ones are used therapeutically, and they involve in defense against other microbes. And as we said earlier, they are more like the voices of the cell, so they help in the communication. We have several subtypes. We have an interferon A, interferon B. We have interferon 2, which is interferon gamma, which is present in our cells. So let's put in an illustration how an interferon actually works. So now let's say this cell is being attacked by the virus, and now the virus enters, is making more of itself and all things like that. Immediately, these cells have the interferon gene, which secretes in the interferon proteins, which secretes the mRNA, is converted into interferon proteins. Now the protein is being excreted out of the cell. And then basically the voice or the interferon, the message is being sent to the nearby cell like, hey, I'm being attacked. Then immediately the nearby cell secrete antiviral proteins, which will basically blocks the further infection of this virus, these cells. So it's just telling, hey, there's a new infection coming on. So you have to protect yourself. So it secretes antiviral proteins, protecting itself from further infection. So that's basically how this is basically one example of how interferon is being put into action. So the third one that we have to talk about is the complement system. And then complement, as is in its name, it's a system or a complement mechanism that actually complement or complements the activity of the immune system. We have over 30 um, complement proteins being seen. They are soluble in the blood and they are membrane bound in tissues. They consist of a cascade of reactions. So basically, like this image here, once one complement system is activated, all the rest are subsequently activated as time goes on. So like if you push one of these, all of this will go down. And then once it's triggered, it will cause a subsequent reaction of all downstream mechanism, like the domino effect. So this is a summary on how the complement system work. The first one is done by activating in any of these three pathways. So it can be a classical pathway, which is being done by the activation recognition of antibody. And then we also have the lectin pathway by recognition of lectin, and then the alternative pathway, which is basically the recognition of the bacterial lipopolysaccharide, or the LPS. And all of these leads to the formation of C3 C3, and then the C3 convertase is being split into two. So we have C3A and C3B. C3B will lead to phagocytosis. C3A will lead to inflammation. And further on, C3B will be converted into C5, which will also lead to C5A, contributing to inflammation, and C5B, leading to membrane attack complexes. And there are also other complement pathways which will contribute to C6 to C9, or contribute to membrane attack complexes. But the main goal, you don't have to memorize the whole thing. The main goal is that the complement pathway always results in three major outcomes: inflammation, phagocytosis, and membrane attack complexes. So that is your take home. Three major outcomes: inflammation, phagocytosis, and membrane attack complexes. So now let's talk about the activation of the complement system. The first one is through antibodies. So the antibody recognize and bind to antigens on the microbial surfaces through acquired immunity, leading to the activation of the C3 convertase. This is more of a classical pathway. And we have lectins, which recognizes and bind to sugars, example mannose on the microbial surfaces, also leading to the same thing, C3 convertase activation. And then we have the recognition of lipopolysaccharides, and then which leads the C3b recognizes and binds to the LPS on the surface of the on the cell, leading to C3 convertase, or just leading to C3 convertase cleaving C3. So now C3 convertase has cleaved C3 and the whole pathway isn't activated. So it's gonna result in three major outcome: inflammation, phagocytosis, and membrane complex. So as I explained earlier, C3 is split into two. We have C3A, C3B. This one leads to phagocytosis, this one leads to inflammation, and the list goes on and on. You only need to know C3 and C5. That's all you need to know. So now let's dig into the major outcomes of the complement system as I've been highlighting on. First one is phagocytosis, which is promoted by opsonization, opsonization, basically through C3b opsin. It binds to foreign cells particles which promote the engulfment of the phagocytosis. So as you can see from here, we have C3b present, and then it just helps in the engulfing of them. It just helps in the recognition of the pathogen, and then helps in the engulfing of the whole thing, engulfing of the microbe itself. So C3 is the green. It binds to the microbial cells as an opsin. So as you can see from here, and it just helps in the engulfing. Second one is the inflammation. So C5a attracts phagocytes to areas which have C3A, C5A, and just increases permeability of the blood vessels, induces mast cells to release chemokines. So as you can see, this is your blood vessel, vasodilation happens, and then you just help releasing out of helping the blood cells to be permeable and releasing out all of the white blood cells to come out to the inflammatory side to attack them. The third one is membrane attack complexes. So basically what it does is the complement pathway produces proteins such as C5b, C6b up to 9, and then they just assembly on the surface of gram-negative and envelope viruses, as you can see from here. So now they just assembly on the surface of the proteins, the assembly on the surface of the cell membrane of gram-negative, or also on the capsules of the viruses, leading to lysosome activity. So they just assemble there and just help recruit lysosome and just cut this surface into cut the cell membrane into pieces and just slice it all out. So here is a more complex form of what we've already discussed. I wouldn't waste much of your time trying to memorize this. Like I said, you don't need to know everything. All you need to know is C3 and C5. All you need to know is C3 and C5. Just ignore the rest. This is just how complex the whole procedure is. I won't even talk about this. You don't need to know this. As usual, try and practice this, and when we meet in person, we'll discuss more about this. So please, good luck in your exam on Friday. Remember, none of this is coming in the Friday exam. This is exam 3 problem. This is not exam 2 problem. So do not learn this for exam 2. Just learn this for exam 3. That is after this exam. So hope to see you in class. Good luck with your exams. And please remember that you are allowed to bring a note card, just a page, so that you can help remember what you can memorize during your exam, so you can have some notes with you to help you write the exam successfully. So good luck in your exams and see you in class.